Good afternoon to all. Myself Sandhya. Today we have to discuss about the culture media. Culture media are mediums that provides essential nutrients and minerals to support the growth of microorganisms in the laboratory. Microorganisms have varying nature, characteristics, habitat, even nutritional requirements. Thus it is impossible to culture medium with one type of culture media. However, they are also microorganisms can grow on a culture media at all any condition. These are called obligate parasites. Common ingredients of culture media: peptone is a source of carbon and nitrogen. Next, second one is beef extract, source of amino acids, vitamins and minerals. Yeast extracts, source of vitamin, carbon and nitrogen. Distilled water, agar, it is a solidifying agent. What is defined medium? A defined medium has a known quantity of all ingredients like carbon source and nitrogen source, nitrate, etc. The medium needs in metabolic, nutritional and physiological growth experiments. What is undefined medium? The medium different complex ingredients unknown. For example, yeast extract, beef, various salts and enzymatic protein, potato, dextrose, manganese, agar, etc. What is complex media? Complex media is nothing but it is other than basal media. It has added ingredients to bring the characteristics of microorganisms with unique nutrients. Types of culture media based on consistency of physical state. There are three types of classification. First one is solid medium, second one is semi-solid medium, third one is liquid medium. Solid media, principle of solid media, it is the isolation of bacteria as a pure culture on a solid medium. Robert Koch realized the use of solid media. Agar is used to hardening the media at 1.5 to 2% of concentration. Solid media allows the growth of bacterium as colonies by streaking on the medium. It is solidified at 37 degrees Celsius. It means in room temperature. Agar is an unbranched polysaccharide extracted from the red algae species like Gelidium. Colonies identification is done on this medium. Examples of solid media is nutrient agar medium, Maconing agar medium, blood agar medium, chocolate agar medium. Growth of bacteria on solid medium appear as smooth, rough, mucoid, round, irregular, filamentous and punctiform. Second one is semi-solid medium. This media shows the motility of bacterium and cultivation of micro aerophilic bacteria. This medium has agar at a concentration of 0.5%. In solid media, agar concentration is 1.5 to 2%. Semi-solid media 0.5% or less. It has jelly consistency. Examples: semi-solid media, Stewart's and amines media, Huck and Leyfoson's oxidation fermentation medium, mannitol motility medium. The growth of bacteria in semi-solid appears as thick line in the medium. Third one is liquid medium. The mixed media shows the growth of large number of bacteria. It is called broth. Liquid medium is called as broth. Bacteria grow uniformly with turbidity. The growth occurs at 37 degrees Celsius in an incubator for 24 hours. Liquid medium don't have the addition of agar. It is for fermentation studies. Examples of nutrient media are nutrient broth, tryptophic soy broth, MRVP broth, phenol red carbohydrate broth. Growth of bacteria in liquid media, turbidity is seen at the end of the broth. Second category is types of culture media based on oxygen requirement. Microorganisms have different requirements for growth depending on oxygen requirements. First one is aerobic media and second one is anaerobic media. Aerobic media, in this media It is easy to cultivate microbes on solid media. The growth occurs by keeping the culture in the incubator. It shows the growth of non-fastidious microorganisms. Examples of anaerobic medium are liquid media and solid media containing first peptone water 1% peptone plus 0.5% sodium chloride 100 ml water. Nutrient broth, nutrient agar, nutrient broth plus 2% of agar. Anaerobic media, that's the media cultivates anaerobic bacteria at low oxygen, reducing oxidation reduction potential. Aerobic media contains extra nutrients like vitamin K, hemine, and oxygen that get reduced by a physical or chemical process. This medium is boiled in water bath for force to force out dissolved oxygen and packed with a sterile paraffin. Thank you.